Right. Can you remember the last week we still discussed innate immunity? In innate immunity, the components of the innate immunity, chemicals, cells, together they play inflammatory response. Right? So we can also do it differently. Innate immunity has inflammatory response, and phagocytic cells and other cells and chemicals are important for the inflammatory response. There's no big difference if we learning purpose classify this. It has chemicals, it has cells. Together, they play a big role. That's the inflammatory response. And we discuss two chemicals. One is interferon. Chemical number one is interferon, secreted by cells which are infected by the virus. Right? So infected by virus. You know, if you want to get a good marks in your exam, certain points are very important. So if you take whole syllabus, you know that exam will not ask always unnecessary things. Like any exam, they will not use all unnecessary detail. They don't want to take as a question. There is a general concept in exams. All over the world, the exams are like that. The examiner try to make questions based on the important content. They don't want to take all the small detail. So then you should make a habit. Very carefully, you need to remember the important components. And if you still feel like you can remember certain minor detail, it's fine. But the strategy is we have to really remember the important component. Now, I will give you one example how this is involved. So, can you remember we discussed complement? So let's go to the complement. So I just skip this interferon. You can go back to the recording and you can learn again. They protect the cell. They also try to protect the neighboring cells by doing this mechanism. Number one, inhibit viral replication, inhibit attachment of virus, support phagocytosis. So these are the three main methods used by the interferon to protect the cells during viral infection. Okay, let's move to the complement. Now, complement proteins are again protein, soluble protein in plasma in inactive form. They are in inactive form. But I told you they can be activated. They can be activated during immune response when there is a pathogen, when there is a tissue damage and molecules of pathogens. So here, what I again highlight, number one, they can be activated by antigen of the pathogen bind with antibody, antigen-antibody complex. So this is the first method. The complement system can be activated, binding of antigen and antibody. So I will not go too much detail. This has a name, classical pathway, like all the details are just avoid and highlight the first activation process is antigen-antibody complex. You will learn antibody and binding, all these things later. But at the moment, antigens we learn from the beginning. Antibody, we also learn a little bit. They are produced by the immune system. They are protective molecules. When antigen bind with antibody, it activates complement. Other one is, I said, pathogen itself. Presence of pathogens can activate or damage tissues. That's the second thing. Damage tissues or pathogen. Third one, some of the specific molecules of pathogen. That's a third one. Some of the specific molecules of pathogen can activate. Now, these are the things we should remember. How complement activate? Number one, antigen antibody complex. Number two, pathogen or damaged tissues. Number three, specific molecules of pathogen. After that, I explain you how inactive form of this protein convert to active form. It then activate the C5 inactive form to active form. So this is the inactive form, and it activate this one. It activate finally the final protein is called C5b29. The function of complement is it can lyse plasma membrane of pathogen. That's very important thing. Even it's not mentioned in resource book. This is not mentioned. Lysis pathogen and support inflammatory response. Phagocytosis. Support inflammation, phagocytosis. So these are the things we should remember. Complement, it's in the plasma. How it activate? Number 1, 2, 3. What is the effect? 1, it can directly kill the pathogen, lyse the pathogen. Number 2, increase phagocytosis. Right. So this tiny detail are important. You need to remember under complement protein. How it activate? 3 methods. What is the effect? 2 methods. Now, I take this question for you. Last 2022 and 2023, the last one, the last exam had. This question is asked. Now, consider one thing. At the moment, you haven't learned antibodies. We have to learn antibodies. So we haven't learned immune system immunity yet, but I just wanted to take this example like this. Select the correct statement regarding antibodies. Correct statement regarding antibodies. Now you just keep rest of the things. For example, leave these 4. Don't consider these 4. Just leave this 1, 2, 3, 4, and take only the 5th one. So when you take 5th one, you should be able to distinguish whether it's correct or not. Antibodies, they mean antibodies bind with specific antigen. Okay, if this is antigen, antibody is Y-shaped molecule, bind with antigen to activate the complement system. That's what you learn here. Here, antigen antibody complex activate complement system. So complement system is the complete system. All this system is a complement system. So here it's a complement system. You can consider C3 protein, C5b29. That's all. It's a system. We consider as a complement protein, complement system. I think the word system and protein, we don't want to think too much like this. Oh. Yes. Here they use the word the system, complement system, and we learn as complement protein. So complement protein all together activation is a complement system. Now it is 100% clear now. Antibody bind with specific antigen to activate complement system. So this statement is correct. Now you see, there is an advantage we can take from some of the questions. When they make the questions, somehow they try to make it harder. Examiner try to make some questions very hard, but somehow examiner has a weakness. Examiner could not make it at the end. If we look the correct sentence, when you see the correct sentence, we feel correct sentence is 100% correct, because we try to remember certain information, and we know, okay, the main thing we learn in complement system, there are 3 ways of activation: antigen antibody complex, pathogen, damaged tissue, molecules of pathogen. After that, they activate complement proteins and complement system. This is the main message we learn here. And the main message is summarized in one sentence: it binds with specific antigen to activate the complement system. This makes easy. Even other choices are hard. They are very difficult. But when it has a very easy right sentence, you should not think about too much to select it. Otherwise, you try to analyze each choice too much. Again, you ended up in a difficult situation. When you see the choice is according to your knowledge is exactly correct, then go for it. So 1, 2, 3, 4, we could not analyze yet because we haven't done about antibodies. We haven't done antibodies, but you can see clearly. Sometimes we learn a different place. Is important again to write the correct answer. Okay, so later we'll also analyze 1, 2, 3, 4 for learning purpose. But you see clearly, you can identify answer. It is very clear. I think when you finish your syllabus one day, you have a lot of information behind you. Some of the information you remember very strongly. So when you remember very strongly, if you see that kind of fact, I think directly go for the fact to answer it correctly. Okay, next, we have learned the activation of adaptive immune system. So when the innate immunity activate, it goes on. At the same time, it communicates with adaptive immunity to bring more help. This is done by, in this way, the main players are lymphocyte, T and B lymphocyte in the adaptive immunity. T lymphocyte, they make cell-mediated immunity. B cell, they make humoral immunity. We'll come to that later. Now what will happen? Our story is going to the lymphocyte. Lymphocytes reside in the lymphatic lymph node. So this is the lymph node. So I told you lymph nodes are distributed all over our body, and the lymph nodes are connected with the lymphatic vessels like this. We learn lymph system in circulation. So when capillaries are there, then fluid is leaking out. Some fluid is going back to the capillary, and some fluid still taken up by the lymphatic system and drained into the vein. Again, they bring to the vein. And you also learn another function of lymphatic systems in digestion. So check digestion lesson. Lacteal is a specific lymphatic system in intestine. They absorb fat, basically the fatty acid and glycerol, and process into a packaging vesicles, and they transport to the liver. And if you go back to the circulation, you can find more information about lymphatic, and go back to the digestion. You can also find lacteal, how it is important. Now again, we are trying to learn some information about lymphatic system, lymph nodes. So lymph nodes are they have a bit harder capsule-like things covering, and then millions of cells stay inside their B and T lymphocytes. So they are connected in two ways. One is lymphatic can connect to this, and then this goes back again to the circulation. This end lymph again connect to the circulation. You can check this one at the end. All lymphatics connected to the circulation through veins. Circulation, you check it. That's also important thing about lymphatic, right? So we discussed this. And here I further try to embed a picture of lymphatics and lymph system. When cells of the immune system from the inflammatory side, they bring a cells. What is the cells coming? If you remember, I told you a one cell antigen presenting cell. So, okay, we come to that later again. We revise that as we advance these things. But here, look at this picture. And you can see in our body the distribution of lymphatics, so lymph nodes. So here we can see this area lymph nodes, and here the groin region. That's called inguinal region or groin region. Again, we have a lot of lymph nodes, and largest lymph nodes, large amount of lymph nodes we can find in the digestive system. You can also think why lymph nodes are distributed like this, why they are too much distributed in the inguinal region, why they are distributed too much in the abdomen, why they are too much distributed in the head mouth area. This we can relate where the pathogen enter, and that place is rich in lymph nodes. You know, pathogen can enter through mouth. That's one common place, mouth and nose, pathogen can enter. When the pathogen can enter, they just land to the upper respiratory system, or maybe throat area. If not, they can go to the intestine and stomach-like places. Then here, you can see large amount of lymph nodes to handle the situation. Pathogen can enter from urinary system, reproductive system, and here again, we have a lot of lymph nodes. So the distribution of lymph node is again not for just for no reason. They distributed maybe based on the entry of pathogens are rich, and they get distributed there. And here, if we take this lymph node, it consists of a covering like this, a hard covering, fibrous capsule made out of connective tissues, and then filled with the B and T cells. In the middle, it's filled with B and T cells. So when cells are coming, bring the message from the site of infection, antigen presenting cells, and after that, activated cells can leave from this side to the circulation and the site of infection. Okay, we continue the journey now. We have discussed initial role of adaptive immune system, how it activates. I also selected another question today that you have to give an idea what kind of a question asked and how we handle these questions. We don't want to stay to revise papers or do question, as you can start now. So we have done 1, 2, 3, 5, and 4 is coming up. Like at least you have done a half, close to half. You can start doing questions. Even if you feel certain facts are not learned, I mean, that's not a difficult thing to go and check the information and can learn, right? Now we're going to check what is correct about lymphatic system. Again, we learn a lot about lymphatic system. Now, again, I am telling you, leave these 4. So we'll come to that later. We can revise because we have learned lymphatic system more than antibodies. Like previous question, we haven't learned, but we have done. Now, look at this 5th one. So lymph nodes are mainly composed of a fibrous capsule like this, which is connective tissue, and it is completely filled with a lymphocyte. It's completely filled with lymphocyte. Now lymphocytes are white blood cells. Because we have learned previously, if you go back to the circulation, I classified the white blood cell into neutrophil, basophil, eosinophil, monocyte, and lymphocyte. And lymphocyte again classified into B and T lymphocyte. So you should have a classification of white blood cell. It's easy to understand certain things. It's just only few cells, like 5-6 cells, and how they divide and how they looks like is the main thing you have to remember. Okay. Now, if you look, the last choice, lymph nodes are mainly composed of connective tissue and white blood cells. Then you need to find, is it correct? I mean, you see now, there's nothing wrong because you have learned lymph nodes are small, this much shape, maybe smaller than this, probably like this in real, something like this. When it's really active, it's completely covered with connective tissues and connected with lymph vessels like this. It also has a blood vessels for the nutrition, and everything should be supplied by the blood vessels, and everything packed B and T lymphocyte, white blood cells. You don't see anything wrong with the 5th one when you look at it. And there is another thing, if you notice, if you look at the tiny things we have to decode, right? Tiny things important. Now, why the correct answer is always placed at the 5th place? Because generally, if you are going to answer this question, you're going to read from this way. You're going to read from this way. So I'm not telling it is always true. Otherwise, it's too easy for everyone. These patterns are there, but just get some everything we want to get the good marks we need. Now you're going to start from first one, first choice, second, third, and fourth. When you start to read, your confusion is going up. Because when you read something, you're going to confuse, okay, first sentence, I'm not sure about this, like this, confusion is going up. I think I'm not going to tell you it's a rule, it's always try. If you have to select the correct answer, why don't you make a habit starting from this way? Because when you read the first one, you have a strong feeling, this is correct. Then when you see this correct, you double check the others if they are better answer. But you are not get confused, you have an answer. You are not get panic. You have an answer, you are confident with this one. Okay, this may be the answer, but I will also check 4, 3, 2, 1. I mean, that's a kind of a small trick, but cannot be used always. But everything is important for us, then we can also use every single small step. Now go to the 4th one. Lymphatic system is involved in the absorption of vitamin C in small intestine. In circulation, we have learned this as fat. Lacteal absorb fat. Lacteal is a lymphatic system in the intestine, it absorbs fat. So then vitamin C is not correct. Composition of lymph is the same as blood plasma. It's wrong because you understand this is the plasma, which is in the capillary. Then the plasma is leaking out to the interstitial space. So everything is not going out. All the proteins are not going out. So it's not wrong because plasma has more proteins, but here is less proteins. And plasma might have more other component like a hormone or like other things more. They are less here. So plasma and the lymph composition is different because plasma is the one form the lymph. So it goes out, the liquid goes out from the plasma, and this is connected. This is collected by the lymph. So the fluid, excess fluid is connected, and it is not the same as plasma. Now lymph drains into the arteries at the base of the neck via two large ducts. Now I reiterate, lymph vessels connected to the veins. At the end, it connects us to the veins, not the arteries. This is wrong. Lymph connected to the arteries. There are two arteries. This one is the lymphatic duct, right lymphatic duct, and thoracic duct. So these two ducts, let's say like this, right lymphatic duct is this, and this is going to connect to the artery. Sorry, vein, right? This is the vein. This is right lymphatic duct. On the other side, thoracic duct, like this, it is connected to the other side of the vein. This is called thoracic duct. Right lymphatic duct, thoracic duct. So they're connected to this vein, and it isn't connected to the arteries. This, I think, we highlighted again in the circulation lesson. And again, we can learn it here. Lymph vessels differ from arteries due to absence of valves. Arteries, lymph vessels differ from absence of valves. Now, when it comes to the valves, vein and lymphatic vessels have valves. Arteries, they do not have valves because if you look the drainage of lymphatic, so they should go up like this direction. Just like a vein. Vein is also going this direction. And we learn in circulation, the pressure in the veins are very low when it starts from this place. Then how does the vein take the fluid blood up? If the pressure is very low against the gravity is this direction, there's no pressure in the vein to bring the blood to the heart. Same story is true for lymphatic. Lymphatic also drain like this direction, and it has very low pressure. So they use a method called movement of the muscles, help them to drain, because I think I mentioned this in circulation. They have a valve like this, and this valve open only one direction. So when the muscles around, let's say this is the muscle contract, this can cause a squeezing of the blood vessels, which move fluid upward. When it moves upward, the valve is closed. The valve is not open this direction. It is not open this direction, it's only open one direction. So this method, check it again, circulation. You have learned this, both lymphatic system and vein, they use the valves. So it is incorrect. It has valves, lymphatics have. So now you can 100% sure your answer. You found these 4 choices are wrong, and you know this one is correct, so you can give the answer. Okay, 2022 again. I select another question because we did the circulation. Only the issue is I don't know how you can remember the last lesson of circulation. If you can't remember, you can revise the blood groups. So this question is asked from the blood group. And this question is quite straightforward. If you know, if you can remember the theory, its answer is clear, because I'm not going to revise it again. You can watch these videos. But I told you, red blood cell has antigen and a plasma has antibody. So if this antigen is A, red blood cell has A antigen, and this person, plasma, has B antibody. So there's no chance of binding it. This will not bind. This is not compatible to A. Antibody is not specific to A. But if this person is taking a B group, so let's say if this person is taking a red blood cell from B blood donation, he got a B blood group. Then this B can bind to the B red blood cell, because B has B antigen. B red blood cell has B antigen, B antibody can bind. Now, if you're smart enough, if I ask what will happen, this is the antigen of B, and this antibody bound to the antigen. Antigen-antibody complex. So antigen-antibody complex. Remember what happened? Antigen-antibody binds. Activate complement. Complement system activate. Complement system can lyse this cell. You know how to do, how it's doing, because you go to the complement lesson and check it. Activate complement system, lyse the red blood cell. So it's not compatible. So anti-B antibodies. So anti-B antibodies present in person A. So I told you, if you take red blood cell B, now B has a B antigen, and that person has an anti-A antibodies. Then there's no binding. No binding, nothing will happen. So if you take A and B, so A and B, they have both A antigen and B antigen. As a result, they don't have antibodies. Otherwise, it can bind and destroy the cells. So naturally, person with AB blood group, they don't have a antibodies. When you take next one, the last O, so O do not have an antigen, neither A nor B. The person has anti-A, anti-A, anti-B antibody. The O person has both. Now you see, this lesson is a revision, right? So you learned this lesson previously in more detail. Now I just quickly revise. Now, if you look at here, this is the anti-B antibody are present in blood group A. And here, blood group O. So then this is the correct answer. A and O, they have anti-B antibodies. You don't want to remember all these things, only you remember A and B antigen. Based on that, antibodies are present. You can draw this picture. You don't want to memorize by heart, remember these things. You can quickly draw this picture and get the information back again. If you know only A antigen on red blood cell and that person has plasma B antibodies. If you know only this first fact, you can draw these pictures and get the information. But this lesson in detail we revised. I just want to say directly from the theory, the question is asked. So if you know it, then you can get the answer, right? Okay, so we continue. The lesson we did last time. Coming to that question. Okay, I take this now. Now lymph node, we get a B cell and T cells. And I told you the antigen presenting cells, they are coming from the site of infections. What they bring, you can see here, they bring antigen. They bring antigen. Antigen is a piece of protein, piece of polypeptide. And this antigen attached to a MHC protein. So we mentioned just for the learning purpose, import some MHC, otherwise it's very difficult to explain this process without knowing MHC. But we have learned MHC in the previous lessons. What is MHC? And they are on the cell surface membrane proteins. They are important for the identification and self and non-self because MHC is unique to person. So immune system knows MHC of the person. Like my body, MHC identified by clearly immune system cells, and they do not make any reactions. So that's identification is important for the immune system to find the validity of this presentation. That means when the cell come and present antigen, they also check whether this is from the system, self, from the self. So then I mentioned you the T helper cells. Try to identify this antigen with MHC, and when it bind, this is a T cell receptor, right? So this is T cell receptor. So T cell receptor bind with the antigen, and then it activate. So I mentioned how this activation process is, there is a protein called cytokines produced by the T helper cells. These cytokines act on T cells